Hey everyone. So when you get a vaccine, the idea is pretty simple, right? It does its job and then it goes away. But what if the instructions it carries stick around for months? A brand new scientific paper dug into that exact question, and today we are going to unpack what they found.
First, let's just start with a number. A huge number. 13 billion. That's roughly how many COVID-19 vaccine doses have been given out all over the world. We're talking about one of the biggest public health campaigns in human history. And it all started with a tiny piece of code.
And that massive global number brings us down to a really tiny, microscopic question. We know the vaccines work to train our immune systems, but what happens to the genetic instructions, that mRNA, after they've delivered their message? Do they just vanish in a couple of days, or do they hang around? That's the mystery we're getting into.
Okay, so before we dive into the nitty-gritty of the study, we have to rewind a little bit. Remember the context. The whole world was grappling with the pandemic, and there was this urgent, desperate need for a solution. And that urgency led to the incredible and incredibly fast development of new vaccine technologies, including the mRNA vaccine from Pfizer BioNTech, also known as Comirnaty.
Now, the core idea here is actually pretty brilliant. The vaccine gives our body a piece of synthetic messenger RNA, or mRNA. This isn't the virus, not even close. It's just a recipe. It teaches our cells how to build a harmless little piece of the virus's spike protein. Then our immune system spots this new protein, flags it as an intruder, and learns how to fight it. That way, it's ready for the real thing.
So, the immune response part, that's pretty well understood. But what's happening behind the scenes, with the vaccine components themselves, is still being mapped out. And one of the biggest questions has always been, exactly how long do these synthetic instructions from the vaccine stick around in our bodies? And where do they go?
We already had some clues. An earlier study had found vaccine mRNA hanging out in lymph nodes, a key part of your immune system, for up to 60 days after a shot. But this new paper, they wanted to look further. They asked, what if it's lasting even longer? And what if it's in other places? So they decided to look in blood, placenta, and even in semen.
And that brings us to the hunt itself. To answer this, the researchers needed a tool that was sensitive enough to find what could be just unbelievably tiny traces of this mRNA, potentially months and months after the shot was given.
So who is actually in this study? It was a small, very specific group of 34 people. This included pregnant women, so they could check the placenta, and men from a fertility clinic to see if the mRNA could be found in semen. It's not a huge group, but it was designed to answer some very specific biological questions.
Now, to find these tiny traces, you can't just use a standard lab test. It wouldn't show up. So they needed something way more powerful, kind of like a genetic magnifying glass. They used a technique called nested PCR. The best way to think about it is like using a super zoom on a photo. It's a two-step process designed to find the faintest possible signal, even if there's only a single molecule to start with.
So here's how it works. In the first round, they take the sample and amplify a big chunk of the genetic code they're looking for. Then, and this is the nested part, they take that product and zoom in, amplifying a much smaller piece that's inside the first one. By amplifying it twice, you get this massive signal boost that lets you find clues that would otherwise be totally invisible.
All right, so the researchers have their super sensitive detection method fired up. What did they find? Let's get right into the results exactly as they are laid out in the paper. Using this nested PCR technique, the study reported detecting the synthetic vaccine RNA in a few different places. They found it in the blood of vaccinated participants, in placenta tissue from the vaccinated pregnant women, and in a few cases, they found it in both sperm cells and seminal fluid.
So the big question, how long did it last? Well, in the pregnant women they tested, the persistence was pretty significant. As you can see here, even after 200 days, we're talking over 6 months later, the study reported that about half of the women still had detectable traces of the vaccine mRNA in their blood. But then, the study found something they did not expect at all. They ran the same super-sensitive tests on 6 unvaccinated pregnant women, and they reported detecting vaccine mRNA in 2 of them. The paper is really clear about this. They don't know why, and the source of this finding has yet to be investigated. A total mystery.
And what about the male participants? The findings pointed to a similar kind of persistence. The vaccine's mRNA was detected in both sperm and seminal fluid, and in one case you can see it was still present in sperm cells 168 days. That's nearly 6 months after the last dose.
Okay, so those are the findings, but, and this is a really big but, what does it all mean? The most important part of understanding any scientific study is putting the results in context. And thankfully, the researchers themselves help us do that.
The paper offers up a possible explanation for why this synthetic mRNA might last so much longer than, say, our body's natural mRNA. The authors suggest that the way it's designed, wrapped up in that protective fatty bubble and with a modified code, gives it enhanced stability. In other words, it seems like it was built to be more durable.
Now, this next part is absolutely critical to understand. The authors are very upfront about what their study doesn't tell us. First, it was a really small group of people. Second, the test is not quantitative. It can say, yes, it's here, but it can't tell you how much is there. Is it a lot or is it a tiny, tiny amount? We don't know. And crucially, they don't know if they're finding the full working recipe or just tiny inactive fragments. And of course, there's still that big unexplained finding in the unvaccinated participants.
So, where does this all leave us? Using a hypersensitive tool, this paper reports finding traces of vaccine mRNA lingering longer and in more places than was previously documented. It's a fascinating new piece to the puzzle. But like all good science, this doesn't really end with an answer. It ends with a much better, more specific question. This study found lingering traces. The next big question for the scientific community is, what, if anything, does that actually mean?